Alright, hello everybody. This week I'm going to be talking about the nervous system and muscle tissue. That's what we're going to study in lab this week. So let's start with nervous tissue. This is a section of the cerebrum of brain. So this is the cerebral cortex. The outside of the brain is composed of gray matter, where you have cell bodies of neurons, and deep to that you have white matter containing mostly myelinated and unmyelinated axons. This particular section has been stained with a stain called fast blue that stains the white matter blue, and the outer portion is basically stained with an H&E-like stain. So here's a high magnification view of the gray matter of the cortex, and here you can see a number of different cell types. The one cell type we're going to focus on in the cerebral cortex this week is this type of large multipolar neuron, which are called pyramidal cells or pyramidal neurons. You can see here they get their name because of their roughly triangular or pyramid shape appearance. These pyramidal neurons usually have large visible nuclei. Here you can see the nucleolus of a nucleus and a little bit of the nucleoplasm around that. If you look carefully, you can see that there are two different colorations in the cytoplasm in some of these neurons. So the cytoplasm is kind of granular and eosinophilic, is typical for cell types, but there's also this deeper sort of magenta colored substance. This substance is called lipofuscin or aging pigment, so you do need to know both of those terms, and this represents metabolic waste products that accumulate in the neuron over time. Because these cells are post-mitotic, they can live a long time, and they tend to accumulate this aging pigment. So here you can see some more aging pigment in this neuron over here, and some more over here as well. So again, in order to find these neurons, you need to be looking in the gray matter of the cerebral cortex, which is going to be this outer part here. So if you're seeing lots and lots of blue, then you're probably too deep and need to look in the more superficial layer of the cortex. In the gray matter here, you can see some blue structures or some background staining or some other axons that pass through here, but it's not going to be a solid kind of blue color as the white matter is going to be. Here's another image. Here you can see two pyramidal neurons. Here's one on the right, here's one on the left. They don't always have that classic pyramid shape. This one here to the left is a pyramidal neuron, but it's got kind of some rounded features to it. You can observe here they have multiple dendrites. There's a couple at the basal surface and an apical dendrite. These pyramidal cells also have axons, which you may or may not be able to see. And you do not need to be able to distinguish between axons and dendrites, except when neurons are stained with Nissl Stain. And that's the only case when you can really distinguish them histologically. So we'll see what that looks like in a few slides. This is a section of the cerebellum, and much like the cerebral cortex, the cerebellar cortex has gray matter, so on the outside, and then you have white matter deep to that. Okay, so it's very similar. The cerebellum is also folded in a series of ridges or gyri separated by grooves or sulci. So here the cerebellum is even more highly folded than the cerebral cortex actually. And so this is all gray matter here on the outside, and the white matter is deep to that. You'll notice that in a H&E stain preparation with cerebellum, the gray matter of the cerebellar cortex doesn't look all the same. You have a lighter layer and a darker layer. There's actually a small layer in between that you really need to look at a higher magnification view to see. So the reason for that is there are three histologically distinct layers in the cerebellar cortex that you need to know. In the cerebral cortex, there's actually 6 layers, but you cannot easily distinguish those using a simple H&E stain. But in the cerebellum, you can distinguish those layers. So let's take a closer look with a higher magnification view at what the cerebellar cortex looks like when it's magnified. So here's a higher magnification view. This is the surface. Okay, so here you can see where the surface is folded. Here's surface over here. It appears on both sides, again, because it's folded. This is white matter, which is the deepest layer. So on either side, due to the folding of the cerebellar cortex, you have the gray matter. And the 3 layers are, here's one right here. Here's the middle layer, which is smaller, and here's the deeper layer. Let's take an even closer magnification view so that I can properly describe those layers. At this magnification, you can really see the middle layer here. So this is a good place to describe these 3 layers. The superficial most layer of gray matter in the cerebellum is called the molecular layer. It has a few neuronal cell bodies. There are some of them, but they're not densely packed together. Mostly this layer contains dendritic processes from a special type of neuron called Purkinje cerebellar neurons, and those are located in another layer. This layer right here, which is called the Purkinje cell layer, contains those Purkinje, the cell bodies of the Purkinje neurons, which you can see right here. These are very large cells, so their cell bodies are much larger than many other neurons in this region. But this is only showing you part of the neuron. In fact, much of the cellular structure of these Purkinje cells is not really visible using an H&E preparation. Their dendritic branches go all the way up through this molecular layer, and that's what most of this tissue is, which is really not visible. It just kind of looks like a solid eosinophilic mass. But in fact, these are mostly dendritic processes of these Purkinje neurons in the molecular layer. So here we have the molecular layer. Here's the Purkinje cell layer, where you have the cell bodies of the Purkinje neurons, and then the deepest layer is called the granular layer, so-called because of the presence of large numbers of granular neurons, which are very small neurons, but that have very distinct basophilic nuclei. And because of the density of them clustered in this area, that's why it has this much more basophilic appearance. So here's an even higher magnification view at the interface between all these 3 layers. So once again, you have the granular layer, here's the Purkinje cell layer, and here's the molecular layer. With this particular slide, you can see a couple of the major dendritic branches extending up to the molecular layer, but as these branches continue to subdivide into smaller branches, they're basically just lost in a tangle of neural tissue. In order to actually visualize the structure of a Purkinje neuron in its entirety, as you can see here, it requires use of a different stain. This is a silver stain or Golgi stain, which can be used to really show the complex morphology of a single Purkinje cerebellar neuron. So the advantage of the stain, obviously, is that it shows a lot more detailed structure of an individual cell, because with H&E, you're only seeing the major parts, the major thick branches of the dendrites in the cell body, but all the rest of it is lost. The disadvantage of this method is it's much more expensive and labor intensive to produce, and it's very selective in staining, so it tends to only show a few neurons and not all of them. So if you were attempting to count the number of neurons or get an idea of cell density, this method isn't the best one for that. This is a cross-section through the spinal cord, somewhere around the thoracic level, probably, and this is a magnified view of the spinal cord here. Again, this is stained with that fast blue stain, which is going to stain the white matter a kind of bluish purple color, and the gray matter is a lighter more eosinophilic color. So note that the distribution of white and gray matter in the spinal cord is approximately inverted relative to the brain, including the cerebrum and the cerebellum. In other words, in the brain, the gray matter is on the outside, the superficial-most layer, and the white matter is deep to that. There are some exceptions we'll talk about later, but generally that's how it's distributed. In the spinal cord, it's kind of the opposite. The white matter is on the outside, more superficial, and the gray matter is deep to that. So this is all white matter that you can see here. And the gray matter that contains the cell bodies of most of the neurons, here you can see. It has several parts to it, or horns, that you need to know. So here, this is on these projections of gray matter on the posterior or dorsal side of the spinal cord. These are called the dorsal horns or posterior horns. Either of those terms is correct. Note that the posterior horns come all the way to the surface of the spinal cord. These are the ventral horns here, and the ventral horns are found on the ventral or anterior side, so you can also call them the anterior horns. Note that they are fatter or wider than the dorsal horns, but they're shorter. They don't go all the way to the surface of the spinal cord like the dorsal horns do. And if you look carefully, you can also see two smaller horns projecting from the lateral sides here. These are called the lateral horns. The dorsal horns contain neurons that are responsible for controlling motor function, such as innervating skeletal muscle. The dorsal horns contain neurons that process sensory information, and the lateral horns process autonomic motor function, such as those controlling smooth muscle and involuntary secretions by glands and so forth. Note that the spinal cord is symmetrical bilaterally, so the left side and the right side look very similar, and the major mass of the gray matter on the left and right side is connected by this bridge of gray matter here. This is called the central gray commissure. Commissures, as you'll learn later, are regions where you have connection between two sides. In the middle of the region of the central gray commissure, you can see the tiny central canal, which is a remnant of the ventricular system of the nervous system that contains a very small volume of the cerebral spinal fluid. So once again, we have dorsal or posterior horn, ventral or anterior horn, lateral horns, central gray commissure, and the central canal. Those are the main anatomical features of the gray matter that you need to know. The white matter of the spinal cord is also segregated into anatomical portions. It's a little bit more complex. That's something we'll really get into in neuro. Here's a similar section of spinal cord, but that's been prepared with a different type of stain, and somewhat confusingly, stains that one of the common stains for white matter stains it kind of a dark blackish brownish color, leaving the gray matter white. So remember that white matter and gray matter get their names based on how fresh specimens look like. So when it's stained, it can change the appearance dramatically. So again, this dark or black colored tissue is all white matter, and the gray matter appears relatively white in this picture. This particular section shows in a bit greater detail some of the important surface features of the spinal cord. The most prominent feature here is the anterior or ventral median sulcus, which is often referred to as ventral or anterior median fissure, because a fissure is a term for a particularly large prominent sulcus, as you can see here. There's also a posterior or dorsal median sulcus along the midline on the other side, the posterior side of the spinal cord, which you can see right here. It's not correct to call this particular sulcus a fissure because it's not considered to be large enough to be a fissure. There are also two sulci at the surface on the dorsal side of the spinal cord where the dorsal horns terminate. So these are called the dorsal lateral sulci. And then finally, in between the dorsal median sulcus and the dorsal lateral sulci on each side, you have these sulci here and here, which are called the dorsal intermediate sulci. So those are all of the anatomical portions of the spinal cord that you need to know for this lab. Let's take a closer look at some of the actual cell bodies of neurons within the ventral horn. And the ventral horn contains multipolar neurons of varying sizes. The largest are these large multipolar motor neurons. By multipolar we mean they have a single axon and multiple dendrites. So this is one type of multipolar neuron, but pyramidal neurons are another example of multipolar neurons that are found in the cerebrum. These neurons, like pyramidal neurons, are very large and have multiple dendritic processes, but unlike pyramidal neurons, they're not pyramid shaped. They tend to be more kind of spherical or regularly shaped. You can also see the presence of lipofuscin, or that aging pigment, in multipolar neurons in this particular section. And again, they have a large prominent nucleus and especially prominent nucleolus. So to find these multipolar neurons, you're going to need, again, to be making sure you're looking at the gray matter. This time in the spinal cord, it's going to be the deep tissue, not the superficial tissue. And these particular neurons are found in the ventral horn. So you can look at the ventral horn on either side to be able to find these large multipolar neurons. This is a section of a multipolar motor neuron from the ventral horn of a spinal cord that has been stained with a particular type of stain which is called Nissl Stain. Nissl Stain is different from H&E in that it stains polyribosomes within the cytoplasm. Polyribosomes that have taken up the Nissl Stain are therefore called Nissl bodies, which are these deep purplish colored structures. These represent polyribosomes, and they are found mostly in the cytoplasm of the cell body of the neuron, right here. You can also find them in the proximal portions of dendrites, but not in the distal portions of dendrites. With a Nissl Stain section, you can actually distinguish between dendrites and axons, because the dendrites do contain Nissl bodies. However, the axon, which you can see down here, does not contain any Nissl bodies. So in this case, you can see the axon because of some background staining here. You can see its silhouette, but it is notably lacking Nissl bodies, so you know this can't be a dendrite. So the axon, as well as the anatomical region where the axon joins with the cell body, which is this wedge-shaped structure right here, that's called the axon hillock. Both the axon hillock and the axon lack Nissl bodies. Nissl bodies, therefore, are only found in the cell body and the proximal portions of the dendrites. You can also see that the nucleoplasm here, there's some background staining, but it's basically missing, lacks the presence of Nissl bodies as well. This is a cross-section through a nerve, so now we're looking at structures associated with the peripheral nervous system, which is composed of nerves and ganglia. So this is a cross-section through an entire nerve, and nerves contain axons that are wrapped in various connective tissue wrappings. So there are three major connective tissue layers or wraps that surround the axons that you need to know. The outermost layer, which surrounds the entire nerve structure here, is called the Epineurium, and the Epineurium is composed of dense irregular connective tissue. So if you look at it closely, you can see lots of collagen fibers and fibroblasts because it's a type of dense irregular connective tissue. Within the Epineurium, you can see several large spherical appearing structures. These are bundles of axons in cross-section, and bundles of axons that are found inside nerves are called fascicles. So here you can see 1, 2, 3, 4, 5, 6 fascicles, and some more over here. So fascicles can contain dozens or hundreds of axons, both myelinated and unmyelinated. Some are smaller than others, as you can see here. Each fascicle is surrounded by another layer of dense irregular connective tissue, which is called perineurium. And so again, if you look at perineurium closely, you should be able to see collagen fibers. There's a third layer of connective tissue, which is called Endo Nerium, and Endo Nerium surrounds individual axons within a fascicle. But in order to see that connective tissue, you need to use a higher magnification. So in this slide, this is a higher magnification view. Here's one fascicle. Here's part of another fascicle that's cut off. Here's part of another one. You can see the perineurium on the outside of this fascicle right here. And inside the fascicle, you can see axons, both myelinated and non-myelinated. The axons in this particular slide appear relatively basophilic, and they seem to be surrounded by this empty white space. That's actually where the myelin sheath would be for these myelinated axons, those that have them. And what happens with the H&E wash is that the myelin gets washed out because the alcohol wash, as we've talked about, removes fats, and the myelin has a very high lipid or fat content. And so that leaves these empty appearing spaces, which is basically an artifact of slide preparation. So here you have the axons in the center that appear to be surrounded by the white spaces, which used to contain myelin. In between the axons, outside of where the myelin would be, you can see this eosinophilic connective tissue. This is the Endo Nerium, and it's a type of loose connective tissue, not dense irregular connective tissue. So make sure you know those three layers of connective tissue that's found in nerves, in the peripheral nervous system. This is an individual axon, myelinated axon, that has been teased out of a neuron, teased out of a nerve. And so this is a single axon, not a whole nerve fiber. This comes from a slide that has been stained with osmium tetroxide. Remember, OSO4 stains fats black, and because the myelin sheath is very fatty, it's really good for staining myelin sheaths of neurons. And you can see right here at this particular location, there appears to be a narrowing or gap where myelin is not present. It's thinned out here. This is an important structure which is called a node of Ranvier, and action potentials will jump from node to node in a type of neurotransmission that we'll talk about in lecture. So this is a very important structure to be able to identify in myelinated axons. Non-myelinated axons, of course, are not going to have nodes because they don't have lipids, or they don't have the myelin sheath to begin with. Here is another slide with axons that have been teased out of a peripheral nerve. Here's a really obvious node of Ranvier. You can really see where the myelin is missing here. You can see some more nodes up here, a little bit more subtle. Okay, so with these particular slides, a lot of the axons are just really clumped together. You may need to find regions where they're a little bit more isolated to be able to see these structures. There is another structure you need to be able to identify on these slides, which are little pockets of cytoplasm within the myelinated axons where myelin is absent. And so, like the nodes of Ranvier, they're kind of lighter in color. So they're similar to nodes of Ranvier, except nodes of Ranvier go straight across. These structures are more of an oblique angle. So you can see them here, and here, and here. So in many neurons, these are at an oblique angle. These structures are called clefts of Schmidt-Lonterman, or you can call them incisors of Schmidt-Lonterman. Yes, you do need to be able to spell that. It sounds crazy, but if you break it down, it's not too bad. They're named after two German neuroanatomists, Schmidt and Lonterman. So it's Schmidt hyphenated Lonterman. You can read about it in your lab manual to get the spelling down. You are responsible for being able to distinguish between clefts of Schmidt-Lonterman and nodes of Ranvier. Note that nodes of Ranvier can also be found in the peripheral nervous system, although these particular axons nodes of Ranvier can be found in the central nervous system, although these particular axons are teased out of a peripheral nerve. But the clefts of Schmidt and Lonterman are only found in the peripheral nervous system. This is another important peripheral nervous system structure. So the nervous system in the PNS consists of nerves, which we've looked at, and also collections of cell bodies called ganglia. So nerves don't contain cell bodies, only axons and connective tissue and some other tissues. But there are cell bodies in some places of the peripheral nervous system, and those structures are called ganglia. Ganglia differ greatly in size and structure, so we're going to look at three different types of ganglia. This is a sensory ganglion called a dorsal root ganglion. These contain the cell bodies of pseudo-unipolar sensory neurons. And dorsal root ganglia, which are part of the dorsal root formation on the dorsal side of the spinal cord. These ganglia are said to be encapsulated, so they are covered with a thick connective tissue sheath or capsule right here that's composed of dense irregular connective tissue, and in some places this connective tissue capsule actually penetrates deep into the ganglion here. So in order to see the cell bodies of these sensory neurons, you need to use a higher magnification view of the inside of the ganglion. So that's what this particular slide is showing. Pseudo-unipolar neurons have large cell bodies, as you can see right here. So like the other neurons we've seen, these are quite large. Again, they have a prominent nucleus and nucleolus, and the nuclei of these neurons tends to be located in the center of the cell. They are surrounded by a type of glial cell, which are called satellite cells. Satellite cells get their name because they're found all around the outside of these pseudo-unipolar neuronal cell bodies, as if they were orbiting it. This is only one place you can find satellite cells. Satellite cells can be found surrounding other types of neurons and ganglia as well. So be able to distinguish between satellite cells and the neurons within a dorsal root ganglion. Now, again, you can see another artifact here. You have this apparent space here between the satellite cells and the cell body of this neuron. In fact, normally this would be filled with the cytoplasm of the neuron, but these neurons tend to shrink back a little bit as a result of the staining protocol, and it leaves behind these little spoke-like structures which are called Neuro Keratin. And again, these are an artifact of slide preparation. These are result from intermediate filaments protruding out of the cytoplasm, and normally that structure would not appear. This is a section of a different type of ganglion. This is a sympathetic ganglion, so these are contain multipolar autonomic motor neurons rather than sensory neurons. So, but like the pseudo-dorsal root ganglion, it's kind of a large spherical collection of ganglion structures. This is frequently referred to as a paravertebral ganglion or sympathetic chain ganglia, because many of these ganglia are attached together and chain-like structures alongside of the thoracic spinal cord. Sympathetic ganglia at low magnification you can distinguish from dorsal root ganglia because although they have a connective tissue capsule as well, it's much thinner, which is this dark structure towards the outside. And I'm not talking about this stuff. This is other connective tissue that has been excised along with this ganglion when it was dissected out. So this is not part of the connective tissue capsule. The capsule here is very thin. Again, looking back at the dorsal root ganglion, you can see how much thicker the connective tissue capsule is. So at low magnification, you should be able to distinguish this from a dorsal root ganglion based on the thickness of the capsule. You can also distinguish it with a higher magnification view because the neurons have a different appearance. So these are multipolar sympathetic motor neurons here. They also have large cell bodies, a prominent nucleus and nucleolus. Note that the nuclei of these cells tend to occupy a more eccentric position within the cell, meaning that they are closer to the edges rather than in the middle. There are also numerous satellite cells surrounding the cell bodies of these neurons, but notice there are not as many satellite cells, and that's because there's really not as much room, because much of the space here is occupied by all the different dendrites and axons from these neurons, so that take up a lot of space. Pseudo-unipolar neurons only have a single dendrite instead of many, and so you don't have quite as much other neural tissue occupying the space here. And we're going to look at a final type of ganglion, which is a parasympathetic ganglion. Now, sympathetic paravertebral ganglia are located adjacent to the spinal cord, very close to the spinal cord. Parasympathetic ganglia are far away from the spinal cord and either very close to or directly embedded within the walls of the tissues that those neurons innervate. So this particular parasympathetic ganglion right here is found in the wall of the gut, small intestines, one place you can find these. And in this particular case, it's wedged between two different layers of smooth muscle. These smooth muscle fibers are oriented perpendicular to each other. So here's one layer of smooth muscle, and here's another layer. You can see the transition right here. Right at the transition point between the two layers of smooth muscles where you can find these parasympathetic ganglia. You can find parasympathetic ganglia elsewhere. This is just one example of them that we're going to look at. Another obvious difference between parasympathetic and sympathetic ganglia, besides their location, is their size. Parasympathetic ganglia are much smaller, as you can see here. And in addition, parasympathetic ganglia are not encapsulated, so they have no connective tissue capsule, whereas sympathetic ganglia have a thin connective tissue capsule. Let's look at this more closely with a higher magnification. Again, you have these large multipolar neurons, in this case parasympathetic neurons, and they are surrounded by satellite cells with an abundance of dendritic processes as well, just like with the sympathetic ganglion. So that's it for the neural tissue. We're going to talk about muscle tissue here. So there are three major types of muscle tissue, and you need to be able to identify all of them histologically. You have smooth muscle, skeletal muscle, and cardiac muscle. Now, smooth muscle is normally found in sheets, and the cells are very tightly connected, and it's usually difficult to see individual smooth muscle cells because they're so tightly connected. But on this particular slide, you have a single smooth muscle cell that has been teased apart from its neighboring cells, and this shows you the shape of these cells. These are the smallest of the three types of muscle cells, and they have a characteristic spindle shape or fusiform appearance, kind of similar to quiescent fibroblasts. In other words, the middle of the cell is large, but they have these narrow tapered ends. Okay, so this is what a single smooth muscle cell looks like when you separate it from the rest. Here's another one up here and part of another one. Normally, though, in tissue, smooth muscle is going to look something like this. Most smooth muscle is organized into sheets. You can see nuclei of many of these smooth muscle cells, but trying to actually distinguish individual smooth muscle cells from others is a little bit tricky, although if you look at their nuclei, you can count them. They typically have a single nucleus in the middle of the cell. Sheets of smooth muscle are often found in different types of layers. So in most of the alimentary tract or the gut, there's a thick wall of smooth muscle, and there are two different layers that are oriented perpendicular to each other. So here's another example of a parasympathetic ganglion surrounded by one layer of smooth muscle in cross-section. Here's another layer of smooth muscle in longitudinal section. And that's because most of the gut, you have two different layers of smooth muscle, an outer longitudinal layer and an inner circular layer. There are some exceptions, such as in the stomach, where there's actually 3 layers of smooth muscle, and we'll look at that in more detail when we explore the alimentary canal after the first practical. So that's smooth muscle. One final note is that smooth muscle gets its name because it has a relatively smooth appearance, whereas cardiac muscle and skeletal muscle, if viewed appropriately, have very obvious striations to them. And so smooth muscle is non-striated, skeletal and cardiac muscle is striated when viewed in longitudinal section. This is a cross-section through some skeletal muscle, and skeletal muscle is arranged in bundles in a very similar way that nerves are arranged in the peripheral nervous system. So much like how axons are arranged in fascicles within a nerve, skeletal muscle is also arranged within fascicles. And also, like nerves, skeletal muscle surrounded by 3 different types of connective tissue. And so if you understand the organization of the connective tissue layers for a nerve, you can understand it pretty well for skeletal muscle. So here we have several fascicles of skeletal muscle. This is all one fascicle right here. Here's part of another fascicle, here's part of another one, and here's another one, or this fascicle might be part of the same fascicle. It's hard to tell. You can see here where there appears to be some separations between the muscle fibers. These are not independent fascicles. This is just where some of the muscle fibers are kind of spreading out after they've been sectioned. You can see that fascicles are surrounded by connective tissue that you can see right here. And this is called paramecium. And paramecium is composed of dense irregular connective tissue. So paramecium surrounds fascicles of muscle tissue, much like Epineurium surrounds fascicles of axons in a nerve. So Paramecium and Epineurium are very analogous structures. If we had a view of the outside of the muscle tissue, which we don't have a slide of, unfortunately, you would see that it's surrounded by an outermost layer of connective tissue, which is called epimecium. And of course, epimecium is analogous to the epineurium that surrounds the outermost layer of a nerve. And as you might guess, there is a third innermost layer of connective tissue that surrounds individual muscle cells, and that tissue is called Endo Nerium. And Endo Nerium, again, is a loose type of connective tissue that's analogous to the, this is called endomysium, which is analogous to the Endo Nerium that surrounds individual axons. Here's a closer look at skeletal muscle. Here you can see the Endo Nerium at this higher magnification better, which surrounds each of these muscle cells. And it's really hard to appreciate the size of these skeletal muscle cells without having some sense of scale. So hopefully when you're reviewing these on your scope, you'll get a sense of the scale to realize just how large these cells are. These are some of the largest cells in the body. They're huge and they are multinucleated, and the reason for that is because they develop from the fusion of many individual myoblast precursor cells. These cells, unlike skeletal or smooth muscle cells, are not tapered, so they maintain a constant diameter across, and so they are very fiber shaped, and for that reason skeletal muscle cells are often called myofibers. And they're just very large cells, hundreds of times larger than many other cells. The nuclei are found at the periphery of the cells, so because they have many nuclei, you can see these nuclei in almost every single cross-section of each of these skeletal muscle fibers. And you won't find the nuclei at all in the center of the cell. They're only along the periphery, because the center of the cell is packed with lots and lots of contractile protein filaments called myofibrils, not myofibers. Here's a higher magnification view of skeletal muscle cells in cross-section. Again, here you can see the nuclei located on the periphery, these basophilic structures. Here's a cytoplasm filled with the contractile myofibrils. Here's some endomysium surrounding each of these myofibers. So that's what skeletal muscle looks like in cross-section. You also need to be able to identify it in longitudinal section. When viewing skeletal muscle in longitudinal section under appropriate light conditions, you can see how it gets the name striated, or how it's referred to as being striated, because you can see these striations, which are alternations of light and dark bands. You'll also notice the presence of the nuclei again on the periphery, so you won't find them in the middle of these muscle fibers, but rather on the periphery. This time seen in longitudinal section. So make sure you can identify skeletal muscle both in cross-section and longitudinal section. To understand how skeletal and cardiac muscle cells get their striations, you have to know the composition of those myofibrils at a much higher level of detail. And that high level of detail can only be visualized using high magnifications that must be achieved using an electron microscope. Light microscopes can't magnify it enough to see the ultrastructure of these contractile filaments. So this is an image of an electron. This is an electron micrograph of these contractile myofibrils within a skeletal muscle cell. And one single skeletal muscle cell can contain hundreds of these contractile fibers. And here we see, here's a single contractile myofibril, and here's another one here, here's another one down here, here's another one up here. And you can see there are alternations of dark and light regions within this electron microscope. These regions have names that you need to know. These dark bands, which are longer, are called A bands, and the lighter bands, which are shorter, are called, or narrower, are called I bands. So you have an alternation of A bands and I bands. You can see some other structures, such as these dark lines in the middle of the I bands, so the color is not completely uniform, but when you look at it with a light microscope, because of the alternation of the A bands and I bands, you just see it as giving it a simply striated appearance. The reason for the names of these bands is very technical, and it has to do with how these different parts of the myofibrils refract light under polarized light. So A band stands for anisotropic, and I band stands for isotropic. Again, it's technical names. You can read about it in your textbook if you're interested in understanding that in further detail. In the middle of each I band, so bisecting each I band, you have another dark line here. These dark lines are called Z disks, or sometimes Z lines. These are disc-shaped structures that are very electron dense here. They contain certain proteins which help hold this entire each segment of these myofibrils together. Z disks are found at regular intervals, and each of these Z disks defines the boundary of the functional contractile unit of these myofibrils, which is called a sarcomere. So one sarcomere is from one Z disk to another Z disk. So here to here is one sarcomere, here to here is another sarcomere, here's part of another sarcomere, here's part of another one, and so forth. So Z disk to Z disk defines one sarcomere. If you look closely in the middle of the A band, which is this dark colored band here, you can also see some different areas in the center that have different electron densities. So in the middle of the A band, there's a lighter region which is called the H band or H zone. And in the middle of that, there is a darker line which is called the M line. Where do these get all their names? Their names are kind of obscure because they reflect German words, because most of the research identifying these the ultrastructure of skeletal muscle fibrils was conducted by German anatomists and physiologists. So the Z actually stands for zwischen, between, and H stands for helle, or light, because this is parts light colored, and the M line stands for mitten, or in the middle of the H band. So whether the German names help you remember them or not, it's just a matter of sitting down and understanding those. So once again, you have dark bands called A bands, lighter bands called I bands. In the middle of each I band, you have a dense Z disk. In the middle of each A band, you have a lighter region called the H zone or H band, and right down the middle of the H band, you have the M line. You won't be responsible for knowing those structures for the quiz, but you do need to know those for the practical. So this was a electron micrograph of skeletal muscle cells, and you can see mitochondria and some other organellar structures between these myofilaments. In cardiac muscle tissue, the arrangement is very similar, and we actually do have some electron micrographs of these for cardiomyocytes, so take a look at those when you're in lab. This is a section, light microscopy section of cardiac muscle that's been stained with H&E. And cardiac muscle is more similar to skeletal muscle than smooth muscle, but it does have some differences as well. So the structure of the contractile filaments is very similar to skeletal muscle. These cells are also long and not tapered as they are in smooth muscle, but they're not nearly as long or as large in diameter as skeletal myofibers. And another difference is cardiomyocytes typically only have one or two nuclei. So some of them have two nuclei or binucleate, but don't have dozens or hundreds of them that you see with skeletal myofibers. Cardiac muscle cells also tend to branch, so there are many branching points here, and for that reason they don't all sit flat together as nicely as skeletal muscles do, so you can see some gaps between the cells that is absent in skeletal muscle tissue. You can also see these dark staining structures at the junction between the ends of adjacent cardiomyocytes. These structures are called intercalated disks. Okay, so here's one cell, here's another cell, here's where the two ends meet, and here's an intercalated disk in the middle of those. Intercalated disks contain gap junctions and desmosomes, and there's an electron micrograph in the lab that shows these structures in much more detail, and they're actually kind of stair-step shaped structures where you find at an interdigitation between the cell membranes of adjacent cells. So try to find the intercalated disks. That was what cardiac muscle tissue looks like in longitudinal section. You also need to be able to identify it in cross section. Now, students often tell me that cardiomyocytes and skeletal myofibers look very similar in cross section, and they do, but these are not nearly as large as skeletal muscle cells. Also, the nuclei of cardiac muscle cells can be in the center of the cell, so you often find them towards the center of the cell. You will not see that in skeletal muscle. Remember, they're at the periphery. Many cardiomyocytes, you may not even be able to see the nucleus. That's because they only have one or two, so you may not have it in a section. With skeletal muscle cells, you're almost always going to see multiple nuclei within every section because the nuclei are so abundant. So those are all different visual cues you can use to distinguish between those types of muscle cells. And finally, we have an image here of a neuromuscular junction. So here we have some axons traveling in a nerve, and here you can see some branches of axons that are innervating these skeletal muscle cells here. The region where you have a synapse between motor neurons and